Good afternoon. I'll be telling you about something quite a bit different than what you just learned about, in which you learn how neurons fire together in order to create thoughts. I'll be going back way in time and to embryogenesis on the genetics of how our brains are formed, because many of us believe that severe psychiatric illnesses in some cases have their roots in genetic and environmental abnormalities during brain development. I have a disclosure that I am part of a company that's interested in cell therapies for neurologic disorders. However, I will not be telling you anything today that is directly relevant to that company. So the severe childhood disorders and adolescent disorders that we're talking about are autism, intellectual disability, epilepsy, childhood schizophrenia, and probably a teenage and adult onset schizophrenia. While not in all cases, many cases have an increased risk if there is a genetic problem that the child or embryo has. And so it's very important that we begin to understand what the genetic risk factors are in order to understand specific mechanisms that predispose to those illnesses. With regard to autism, work by Matt State and others in the field have recently made big discoveries and have identified literally tens of genes that are increasing the risk for autism. It may be as high as several hundred genes, which are risk factors, individually or in combination. When one looks at the properties of those genes, and by properties I mean what kinds of proteins they encode, it's becoming evident that the types of genes and proteins that are encoded by these disease genes are distributed in many parts of the cell. So this picture, which is a schematic cartoon, shows the parts of the cell where some of these genes that are linked to increasing the risk for these diseases. This is the nucleus of the cell, and shown here are two of the genes that are implicated in autism. These are synapses, and many of the genes that are linked to autism make proteins that are found at the synapse. And then this is a dendrite down here, and this corresponds to a gene that's expressed and functions therein. So there's no simple answer in terms of what part of the neuron, or even what stage of development, and actually what parts of the brain are the essential risk areas. It may be quite distributed. As you heard just now, the thoughts are not encoded by one place in the brain. They are encoded by ensembles of neurons that are interconnected between brain regions that are humming together and creating complex thoughts and behaviors. And therefore, one could disrupt those circuits simply by disrupting one part of the circuit. And so individuals may have different parts of their circuit, the important circuits that are disrupted, leading to the similar behaviors. While many of the genes encode proteins involved with synapse function and other aspects of neuron function and development, my lab focuses on genes that encode proteins that function in the nucleus. The nucleus of the cell, if you followed Woody Allen's type of depictions, would be the headquarters of the cell where decisions are made and are executed by the process of gene expression, which basically means, turning on and turning off genes, which then leads to production or the repression of making RNAs, which then leads to the production of proteins. This is a list of some of the genes that have been recently implicated in increasing the risk for autism. And these genes are all encoding proteins that regulate the nucleus. So perhaps as many as a third of the genes implicated in autism are encoding nuclear proteins. So let's talk a little bit about what nuclear proteins are. The parts of the nuclear apparatus that controls gene expression are largely proteins called transcription factors, which you can think of as on-off switches that turn on and turn off genes. The place on the chromosome where these transcription factors turn off and turn off genes are called enhancers. And the way that enhancers and transcription factors work as an ensemble depends upon the nature of the chromosome at that time, and that the nature of the chromosome, whether it's active or repressed, is controlled by other proteins that as a group are referred to as the epigenetic molecules. So these three types of components in the nucleus are all need to be understood vis-a-vis gene expression and how disease genes are acting. So what my lab has been working on for the last 30 years or so are molecular mechanisms underlying how mutations and transcriptional regulation control brain development. And by luck, some of the work that we have done has shed some light on autism. So let's just go back a little bit for some information. In our nucleus are our chromosomes. Our chromosomes are made up of DNA and proteins. And these are cartoons showing what DNA looks like. And mutations take place by changing the molecular structure of the DNA, which changes the code that it has for encoding the types of proteins that are made. In the nucleus, the DNA is not naked. It is wrapped up in proteins. And this shows a picture of naked DNA then being wrapped up in proteins called histones, which when DNA and histones are interacting, you form a structure called chromatin. So when you take a picture of chromosomes during cell division, you're actually looking at the ensemble of the DNA plus the chromatin proteins. Now, the parts of the genes that encode proteins only make up, I think, about 1% to 2% of our DNA in our genome. And the other 98% of the DNA, until recently, hasn't been understood as to what it does. A lot of it, are regulatory elements, with this jargon, cis regulatory elements, meaning they lie right next to the genes on the same chromosome. And there are two major kinds of regulatory elements that I want to mention. One are the enhancers that I brought up earlier, where this is where the transcription factors are binding and turning on and turning off the genes. And then promoters, which are more of the workhorse of the gene regulation, which is where RNA polymerase binds next to the gene and when activated by the transcription factors, initiates the production of RNA. This is a cartoon of an inactive gene. So here's the gene here. And then this is a loop of the chromosome. And way over here is the enhancer. If the enhancer is not close to the promoter on the gene, then the gene is not active and it's not transcribed. However, if this enhancer is brought next to this promoter, as shown in this part of the schema, so that then you have the enhancer next to the promoter and the transcription factors interacting with the RNA polymerase right at the beginning of the gene, then you have an active gene. So there's quite a bit of gymnastics that the chromosome has to do, bending and interacting with different parts of the chromosome in order for gene regulation to operate properly. So one of the things that my lab has been interested in the last 5 years or so is identifying enhancers. These are, again, the regulatory elements that may constitute a large part of our genome and until recently hadn't been discovered. So let me just, to give you an idea about what we do, show you a experimental paradigm that we use in order to study enhancers. And we do this in mice, and we make transgenic mice, which means that we experimentally add extra DNA into the genome of an individual mouse in order to study how enhancers work. So for instance, we'll make an artificial gene shown up here, which will take a candidate enhancer sequence and put it right next to a gene that will make the cells in the mouse turn blue if the enhancer is active in those cells. And then that piece of DNA is injected into a one-cell embryo of the mouse. And that one-cell embryo then is implanted into the uterus of that mouse and allowed to grow up. And then we can look in the embryos that are derived from these injected one-cell ova. And here's an example of a mid-gestation mouse embryo in which we were successful in identifying an enhancer element that turns the brain blue, as you can see here. But it doesn't turn the brain blue everywhere. It makes the brain very blue right here. And that's in the basal ganglia of the embryonic mouse. So in this way, we can assay at relatively high throughput. My colleagues, Axel Wiesel and Len Pinocchio at Lawrence Berkeley Laboratories, hundreds of these enhancers, and we've identified literally 300 different enhancers that are active in the embryonic brain, some in the embryonic cortex, some in the embryonic basal ganglia, and so on. These are examples of 3 such enhancers. So I'm showing you here are sections through individual embryos, in this case 3 different embryos, that were injected either with enhancer 1538 or 1056 or 799. And you may notice that the part of the brain that is blue in each case is in a related region, which is the basal ganglia of the embryo, but in different parts of the basal ganglia. And so this way we're beginning to crack a code for the types of enhancers that drive expression in the embryonic basal ganglia, but also we're looking for enhancers that are active as the brain matures and even in the adult brain. Another step in our types of analyses are to try to identify the transcription factors that are regulating these enhancers. And one of the ways that we test these transcription factors is we make mice that have a mutation lacking the transcription factor and then assay what is the effect on brain development. So when I was a NARSAD young investigator, when I was still at Stanford, in Roland Cirinello's laboratory, I started identifying genes that were expressed in the embryonic brain. And this actually yielded a very robust set of genes, many of which have turned out to have fundamental roles in brain development. And 2 of them are shown here. One is a transcription factor called DLX2, which is expressed in the basal ganglia, and the other is a transcription factor named TBR1, which is expressed in the embryonic cortex. As it's turned out, TBR1 is one of these high-confidence autism genes that Matt State's laboratory and Nenad Sestan and others have identified. Our work on TBR1 over the years has shown that it is critical for defining the identity and properties of deep-layer cortical neurons. And Bin Shen's lab has gone on to show that if you lack TBR1, the layer 6 neurons are confused and they think they are layer 5 neurons, which has fundamentally bad effects on brain function. DLX2 is one of these other transcription factors we discovered 25 years ago. And unlike TBR1, which is expressed in excitatory neurons of the cortex, DLX2 DLX2 is expressed in inhibitory neurons in the basal ganglia and inhibitory neurons in the cerebral cortex. And DLX and its family members are involved with degeneration of these neurons and in regulating their function. What my lab wants to do over the next, let's say, decade is to decipher circuits in the brain, but not circuits of neurons. We want to decipher the circuits of transcriptional pathways that control the properties of neurons. So just as there's a circuit board in your computers that are interconnected and control how the computer operates, there are circuits in each of our nuclei of our cells where transcription factors with their enhancer regulators control circuits of gene expression. And we are now in the process of deriving what these circuits are. So here is a proposal for a circuit in a GABAergic neuron where DLX1 and 2 are at the top of the pathway and control these other genes. And the reason why I show you this is that once we have these circuits figured out, then we'll be able to see whether mutations that cause disease are sprinkled within the circuit. Because if you disrupt a circuit in any particular place, you can disrupt not only that step, but you can also disrupt steps downstream of it, and you can create replicas of the same problem by disrupting the circuit in different places. So already we have found that in this circuit there are disease genes that cause mental retardation, epilepsy, and in some cases autism, that are downstream of DLX2. So I think the exercise that we're going to be engaged in circuit identification in the developing, and I hope also in the mature brain, will be very helpful for elucidating how the genetics of brain disorders operates. And already what we've learned is through making mutations in these genes, that if you mutate the DLX genes, which are expressed in these blue regions, you disrupt the development of basically all inhibitory neurons in the cerebral cortex and basal ganglia. And we can do this at different levels by disrupting, making more or less severe mutations. And our understanding of how you make inhibitory neurons led Mike Merznick and I some time ago to propose genetic models as well as neurophysiological models of how disruption of inhibitory and excitatory balance can change signal to noise in various brain regions such as the cerebral cortex and lead to inefficient cognitive functioning. So in summary, what I hope to do over the next decade or so, and thanks very much to the NARSAD awards that I have received, including the Ruane Award, is to gather more insights into the molecular biology, genetics, and developmental neurobiology to get deep understandings about transcriptional mechanisms that underlie childhood neuropsychiatric disorders. Thank you.
Thank you. Are there questions for Dr. Rubenstein? Yeah. Yes, ma'am.
Once you understand the underlying neuromechanisms,
Here we go. Here we go.
what will you do?
Ma'am, can you grab this so people can hear? Yeah.
Once you do understand the underlying mechanisms, then what can you do to modify that? And is it something preventative for the future, or is it some type of treatment that you would provide, considering some of these are congenital and some are possibly congenital but don't develop until later ages?
Very good question. Because it's already evident that the genetic complexity leading to risk and disease is high, we have to be very careful that we don't lump together people with a particular disease name as a unitary biological group. What the kind of work that we do, as well as others, is going to provide what's called genetic fingerprinting that will help us subdivide the patients into different groups that have specific molecular lesions. By identifying the genes that are wrong, we can understand proteins and their functions in the cell, which will give us hopefully insights into the more specific biological therapies that will be tailored more efficiently to those individuals. This will take time, and it's not going to be a quick fix, I don't think, but I think it's light that will really begin to lead to true understanding and better therapies.
Great. Thank you. Yes, sir.
Last year, Robert Friedman was here talking about the alpha-7 nicotinic receptor.
Can you, here you go.
Can you hear me?
Yeah, right there.
Last year, Robert Friedman was here talking about the alpha-7 nicotinic receptor and looking back in utero and giving people during their pregnancy phosphatidylcholine supplementation, and therefore providing an increased choline in utero, and is now following these people forward. I just wanted to know your feelings on an approach like that.
I like the idea that understanding the specific molecular lesion that individuals have will give us ideas about therapies. Unfortunately, I'm not educated enough about that specific story in order to be able to give you a good answer about how effective I think they may or may not be.
Yes, ma'am.
Environmental factors after the baby is born
Yep.
can affect the environmental factors after a premature child is born can cause autism?
Is that a question or a statement?
Yes, I'm asking you. Environmental
can I-
factors like excessive heat
Yeah.
or water contaminated.
I believe that environmental factors are very important in definitely in modifying disease strength and perhaps in a disease initiation. However, other than viruses, I'm not that familiar with environmental causes of autism.
Yes. So this is related to the first question. In terms of intervention, there has been some talk about microglial being overactivated because of systemic inflammation.
Yeah.
And added to that, the interaction between the gut microbiome as a hub for neurotransmission and the immune system. So I know that you are targeting specific areas to develop, a more specific approach. But alongside that, do you have any thoughts about the systemic and other types of theories about what may be going on? Thank you.
I'm familiar with some of the very exciting work that's uncovering a potential role of interaction of the immune system outside the brain with the developing brain, and I've seen data presented at scientific meetings that I'm pretty convinced about that suggests that lymphocytes can produce proteins which can cross the blood-brain barrier and can affect brain development. And so I'm looking forward to seeing more progress in this area because it's a new concept, a relatively new concept, and may lead to new insights into mechanisms causing the disease and possible different types of therapies.
Okay. Well, thank you very much, Dr. Rubenstein. Really appreciate it.